Section 5.2, Mitosis and Cytokinesis. So DNA is a double-stranded molecule made up of 4 different types of nucleotides. And remember, a nucleotide is a phosphate, a sugar, and one of 4 nitrogen-containing bases. Those bases are adenine, thymine, guanine, and cytosine. Okay, DNA carries a genetic code for living things, basically the blueprint of life. It codes for everything necessary to have things live. In order for DNA to get separated during mitosis and cytokinesis, which is division of the nucleus and division of the cytoplasm, DNA has to be packaged into something called chromosomes. So for a human body cell, there's 46 chromosomes in each body cell. If you were to take these chromosomes and lay them end to end, all stretched out, our DNA would be about 10 feet long per cell. In order to get this 10-foot long structure into this tiny little nucleus, it's going to have to get packaged in order to fit. So it's going to get condensed and packaged and tightly coiled up in order to fit inside the nucleus. So DNA packaging part 1 is the DNA is going to wrap around proteins to organize and condense. Right now, during interphase of the cell, this is the nuclear membrane, and this is the genetic material, the DNA, just kind of laid out in there. Kind of looks like a plate of spaghetti, not really organized, not condensed. But during mitosis, which is where the nucleus is going to divide, and in doing so, the DNA will divide evenly. It has to get tightly condensed into these chromosomes. The cell's still doing all its normal cell functions, growth, repair, whatnot, but the DNA is starting to condense. So it will actually get visible during this stage of cell division. So DNA packaging part 2, the DNA, these long strands of DNA, remember DNA is this double-stranded molecule like this. Okay, just long, looks like a string. DNA is going to wrap around this protein called histones. Okay, so these are histone proteins, and this is the DNA wrapping around it, kind of like line going on a fishing reel. You could, if you wrap it around there, you can get a lot of this stringy stuff around, or wrapped around this histone protein. Okay, and this thing would be a nucleosome. Okay, the histone protein with DNA wrapped around it would be a nucleosome. When you get a bunch of these long strands of nucleosomes, it's called chromatin. So chromatin would be like this. It's condensing. Okay, if you were to take a whole bunch of these nucleosomes and kind of put them in this structure, that would be called chromatin. Okay, so what this structure is right here is just this part on this long strand of DNA. Okay, when the chromatin coils up in like a telephone cord, it's called chromatid. Okay, so these two structures would be chromatid. And the 2 chromatids, which would be identical and are attached at the center, these 2 chromatid strands would make up the chromosome, and they're held together in the center at something called the centromere. Now, the reason we're getting these identical chromosomes is DNA is already replicated. Okay, it replicates in S phase of the cell cycle, and it's replicating to get an identical genetic copy. So when the cell divides, each new cell gets exactly identical genetic material. You can see this entire thing. This is on page 267 of your book. Okay, and if you click on that thing, it'll animate this whole thing so you can see it with some more definitions for it if you like. Okay, at the very end of the DNA molecule, are things called telomeres. Telomeres are nucleotides that don't code for any genes. They don't code for proteins. They don't code for anything important to development of the cell. And what this does is it helps prevent the loss of genes when the ends of the chromosomes accidentally attach to each other. Every time DNA is copied, a short section of this telomere, the end is going to get lost. Okay, and so since this end is coming from the telomere, which doesn't code for anything important to the cell, no genes, no genetic material that's actually necessary is lost. So the telomere, like this, is just the very end of the chromosome. And again, it doesn't code for anything important to the cell. It's just some excess genetic material. So when the cell, or when the chromosomes divide, they don't lose anything vital to the cell. Okay, cell division in eukaryotic cells. Okay, it comes from two major phases: mitosis, which is division of the cell's nucleus and the contents of the nucleus, which would be all the genetic material, the DNA. This is going to happen during asexual reproduction. Asexual reproduction is when a cell just divides to make another cell. When a cell is growing, or when an organism is growing, so it's getting more cells to make up the organism, or when cells get damaged and need to be repaired or replaced. The second part of this is called cytokinesis, which is division of the cytoplasm. At the end of cytokinesis, two genetically identical daughter cells would be produced. They're called genetically identical because they have exactly the same number of chromosomes, which are identical copies of one another. Okay, and mitosis is made up of 4 phases: prophase, metaphase, anaphase, and telophase. And cytokinesis would be division of the cytoplasm, which is going to divide this into 2 identical daughter cells. So stages of mitosis we're going to go through in detail. Again, there are 4 stages of mitosis. We'll go through each one in the upcoming slides. The first stage is prophase. Okay, during prophase, the chromatin, remember chromatin is the DNA wrapped up around the histone proteins. It's going to condense, and the chromosomes will be visible under a compound light microscope. The nuclear envelope, which is the nuclear membrane, is going to start breakdown. It's showing that this is starting to break down, dissolve, getting out of the way. And something called the spindle fibers, which is part of the cytoskeleton, are going to start growing from the ends of the cell. And eventually those will move all the way across and attach to the chromosomes. So in prophase, we have chromosome thickening and getting visible. Chromatin condenses. The nuclear envelope is going to start to break down, dissolve away, and the spindle fibers will start growing from the ends of the cell, at both ends of the cell. The second phase is metaphase, where the chromosomes line up along the equator. The equator is just the middle of the cell. And the spindle fibers, which have been forming from the ends of the cell, will attach all the way to the middle of the chromosome, which is the centromere. Okay, so meta means middle. The chromosomes are lined up on the middle. And at this point, the nuclear material, the chromosomes, are getting ready to divide. The third phase is anaphase. Anaphase, the spindle fibers, these fibers are starting to shorten, and the sister chromatids are separated. These chromosomes are moving to this end of the cell, and these sister chromatids are moving to this end of the cell. As you can see, the cell is kind of like elongating. It's getting stretched out. It's moving this direction. Okay, and as these things get pulled the opposite poles by the spindle fiber shortening, the division of the nuclear material, this will end up being in one of the new cells, new daughter cells, and this will end up being in the other daughter cell. The fourth phase of mitosis is telophase. In telophase, everything that happened in prophase is going to happen in the opposite direction. So the nuclear membrane, which is the membrane surrounding the nucleus, will begin to reform. Okay, the chromosomes, which were condensed and very visible, will begin to unwind or uncoil. So they will start going back to that spaghetti-like shape. And the spindle fibers, which form from the end of the cells, will dissolve away. And at this point, we almost have two new cells, almost have two new nuclei in these cells. After mitosis comes cytokinesis. Cytokinesis divides the cytoplasm and everything in it, the organelles, the cytoplasm, everything, evenly between the two new cells. And it's slightly different in plants and animals due to plant cells having a cell wall. In animal cells, this would be an animal cell on the top, membrane forms. Okay, this is a belt of proteins that are going to kind of pull these parts together and pinch this nuclear membrane in to eventually form two new cells. In plant cells, a cell plate forms due to cell wall material, the cellulose that makes up the cell wall, being brought to this area between the two new nuclei, and it's going to actually build up a cell wall between them, because these vesicles that are bringing in this cell wall material to literally cut the cell in half and form two new plant cells. These guys are still called daughter cells. So it's slightly different. In animal cells, it's formed by this belt of proteins that pull the cell membrane in and literally pinch it in half. And then plant cells, this cell plate forms between the two new nuclei to form the two new daughter cells. Okay, so for the next couple of slides, you're going to look at first some diagrams of cells going through the process of mitosis and cytokinesis. And from looking at that, you're going to figure out which stage of mitosis they are in. Okay, so for this diagram, what we're seeing is it looks like two cells, right, that are being formed. It looks like the nuclear membrane is starting to reform, right? The chromatid or chromatid material is starting to unwind, and the spindle fibers are going away. So if we have two cells and all this kind of happen, this cell would be in telophase, right? The nucleus is already divided. It's starting to pinch in the cleavage for forming, so this would be in telophase. Okay, so for this one, there's only one structure. The chromosomes are very visible. The nuclear membrane is still kind of breaking down. The spindle fibers are forming. So in that scenario, the second diagram, this cell would be in prophase, and you can tell that from what the genetic material is doing. The chromosomes look like this. The spindle fibers are forming. The nuclear membrane, this white material, is just starting to break down, so prophase. In this one, you can tell by the shape of these guys that they are being pulled this way and this way by the spindle fiber shortening. So since they're being pulled to opposite ends, this one would be in anaphase. Here, one of the easiest ones to see, all the chromosomes are lined up on the middle of the cell on the equator. The spindle fibers are formed, the nuclear membrane is gone, so this guy would be in metaphase. Okay, so on this one, we're going to get, or the next couple slides, we're going to get actual electron micrographs of cells, and then looking at these real cells, we'll determine which stage of mitosis they're in. So here, looking at the chromosomes, you can see that these are kind of bent. You can still see spindle fibers here. It looks like the cell is being pulled in this direction, or the genetic material is being pulled in this direction. Okay, so this one would be in anaphase, and you can tell that by what the chromosomes look like and how the cell is starting to elongate and pulling everything towards opposite ends of the cell, pulling all the genetic material towards opposite ends of the cell. In this one, we can see two cells that have formed, or two nuclei that have formed. The chromosome material, the genetic material is starting to dissolve. Okay, since we have two areas of genetic material, this guy would be in telophase, because the nuclear material, the DNA is already divided, it's starting to form this cleavage furrow, it's starting to start the process of cytokinesis. We have two cells, so telophase. On this one, we just have all genetic material right in here. Okay, these spindles are starting to form. I don't see a nuclear membrane, so this would be in prophase. For this one, all the chromosomes seem to be lined up right in the middle of the cell. When the genetic material is lined up in the middle of the cell, it would be in metaphase.